Alright, so what we're looking at right here is just a fun little experiment going back to the microbes around us. So what we did in this experiment is we took a coffee and a water that were both relatively old. The coffee was sitting out for about a month or two in a closed container. And then we also added water. And these are both on TSA plates. So TSA nutrient auger. So what we wanted to do here is to kind of look at what microbes might be growing in each environment. Okay, so recently there's a study published that not washing your water bottle could be a great source of salmonella and other microbial disease or infection. So seeing this, after noticing that I myself had left my water bottle sitting with water in it for a few weeks, I thought it'd be an interesting little experiment to look at. So what we did here is we plated 100 microliters of both the coffee and the water. So you can see here we have a lot of growth bacteria on our water. Okay? So the main takeaway from this, microbes around us, make sure to sanitize as best you can. Clean your water bottles instead of just reusing them without cleaning. Also, this also shows how robust our immune system might be. So this is something that would be covered in other courses concerning microbes or even your immune system in its own course itself, showing basically how the body can handle millions of microbes at one given time. So other studies have shown that the amount of human cells and bacterial cells in the body are approximately the same. The only time that there's more human cells than bacterial cells is either when someone's undergoing an antibiotic or treatment or after defecation. So that's another time where, you know, your innate human cells, you'll have more human cells than bacterial cells. So the coffee. Now this is something that we're expecting to see a lot more of. So this is an interesting result. It could have been that really the main possibility in this plate is that maybe this TSA plate didn't have the correct nutrients to supply to whatever microbes was growing in the coffee. So when we plated this, we saw thin like biofilm of microbes when we plated. So this just shows that there was probably microbes in the coffee, or I would almost, I'd bet my $5 in my pocket that there were. However, these were not optimal growth conditions for the microbes that we had on TSA plates in 37 degrees Celsius.